All right, now we heard about 3 really awesome projects conducted at the Center for Sustainable Development, and we will switch now a little bit to projects that were done at the Cape Eleuthera Institute, more about marine ecology topics. My name is Tanja Schwank. I'm a grad student from Germany conducting my master's thesis research here at the Cape Eleuthera Institute, and I had the pleasure to be one of the advisors of the Stingray research class this semester.
Hi, my name is Casey Sullivan. I'm a research tech here with our Elasmobranch program. I also co-advise this wonderful group. This project is actually a continuation and is going directly towards 2 master's students at the University of Exeter, which Dr. Owen O'Shea, who's the head of the Elasmobranch program, is supervising. So this is really exciting and really amazing research, and we would love to introduce our group. So thank you so much, Tanya and Casey.
We are the Spring 2017 Stingray Research Group. I'm Grace Crenshaw.
I'm Lucia Bowles.
I'm Patrick Howard.
I'm Grace Fogarty.
I'm David Keene.
I'm Luke Weinstein.
And I'm Katie Grogan.
And this semester we've been studying the diets of 2 species of stingrays here in Eleuthera. But before we got into our research with you all, we first thought it was important for you all to understand the importance that these rays have in their environments here. So these rays are mesopredators, meaning that they sit in the middle of the food chain, acting as regulators between both the upper levels of the food chain above them, the apex predators, like sharks here in Eleuthera, and the lower levels of the food chain, like your primary producers. So these rays also are considered ecosystem engineers, meaning that they have both a physical and chemical impact on the environment here. So physically, these rays are, when they go through their feeding process, they stir the sediments on the ocean floor looking for prey. So they're able to physically change these environments that they're in. And also chemically, they provide then oxygen into those deeper levels of the sediments that were anoxic before them. So they're then able to provide different microhabitats for organisms to dwell in.
So for our project, we had 2 subjects. We have the southern stingray, Hyponus americanus, and the Caribbean whiptail ray, the Styrochirus smarte. As you can see, the southern stingray has a more diamond-shaped disc, and the Caribbean whiptail has a circular disc. That's how we distinguish these 2. As they're both so ecologically similar, the question arose, how are they coexisting in the same area without outcompeting each other? So that leads us to our objectives. As these rays are data deficient, we don't know much about them, so we wanted to describe their dietary contents and then analyze the intra and interspecific between those. This would lead us to understand the role that they play in the larger ecosystem. For our project, we had a couple study sites around Cape Eleuthera, as well as one up in North Eleuthera at Bottom Harbor. We are looking at primarily sand flats for the southern rays and creek systems for the whiptail rays.
The capturing process for stingrays begins with spotting for dark spots moving in the water. Once it's identified as a ray, the team moves towards it and begins to encircle it. A gap is made in the circle that's covered by a seine net. The ray is herded into the seine net and secured with black dip nets. Once the ray is in the dip net, the team immediately moves to cover the barb with wet cloth and Velcro straps to make sure the barb is not of any danger to the team or the stingray itself. Given the amount of time it takes to get our hands on a stingray, we try and take as many measurements and samples as we can to help with other projects at CEI. The most important measurement for our team is disc width, which is used for assessing ontogenetic phase in southern stingrays, or whether or not it's a juvenile or a mature. The most important sample for us is stomach content, which is achieved through gastric lavage. Gastric lavage is an invasive yet non-lethal method of extracting the stomach contents of elasmobranchs. A 60-millimeter syringe is filled with seawater, connected to a silicone hose, and fed through the buccal cavity, or the mouth of the stingray, into its stomach. Once it's there, seawater is pumped through the syringe, the hose is slowly removed, and the stomach is firmly massaged, and the ray will most likely achieve regurgitation onto the lavage tray, where the samples are collected for later analysis in a lab. Any gut contents that are also excreted from the cloaca are also saved for analysis at the lab.
So our final step is unwrapping the barb from the cloth and removing the Velcro straps, and then releasing the ray back into the ocean. So once we've collected all of our samples in the field,
We go back to the lab and begin by visually analyzing the stomach contents samples to identify prey taxa. We count the taxa that we've identified as well as weigh the sample. We weigh the unidentifiable matter as well that has been too digested for us to be able to identify. So we use 3 main metrics in our study, the first being %N. %N is numerical importance, which tells us the amount of prey items within a certain prey taxa in the stomach sample we've collected. Next is %M, or mass. This tells us the gravimetric contribution, or simply the mass, of a prey taxa. Next, we have %FO, or frequency of occurrence. This essentially tells us how frequently a prey taxa occurs in the diet of a stingray. We also calculate IRI, or the index of relative importance. The index of relative importance is important for us because it addresses any possible biases that might occur by using only one of the previously mentioned metrics, as IRI encompasses all 3 of them. And it tells us the relative importance that a certain prey type has in the diet of a stingray.
And so for the results of this study, we lavaged a total of 75 rays, and out of those 75 rays, 47 of those rays regurgitated, while 28 of them had empty stomachs. And so right here we have a prey accumulation curve, and what this shows us is on the x-axis down here, we have the number of stomachs, and these stomachs are randomized, and the number of prey taxas we found in the rays. And so on the y-axis, the new prey taxas we identified, it goes up each time. And so we hit on 9 stomachs right here. We had an asymptote of 11 new prey taxes. For the southerns, for 13 stomachs, we had an asymptote of 13. And for the southern juvenile and mature, juvenile being the blue, mature being the red, for 12 stomachs for the juvenile, we hit an asymptote of 11. And for the mature, we hit an asymptote of 13 on 9 stomachs. And the prey categories we used for this project were crustaceans, worms, teleosts, and mollusks.
So using that metric that Grace addressed earlier, we can see for the diets of the southerns that 74%, including the 46% shrimps, 11% crabs, and 17% crustaceans that were too digested to allocate towards a specific prey group within crustaceans, makes up a total of 74% crustaceans. There were also 10% teleosts, 15% worms, and 1% mollusks. Mollusks were very insignificant, as you can see, but we thought it was interesting that we found them only in the diets of the southerns and not the whiptails. Now for the diet of the whiptails, we can see that 63% was made up of worms, while the other 37% was made up of crustaceans, the majority of that being 36% shrimp. Teleosts were so insignificant in this diet that they don't even show a figure above 0%, but they were found. And again, mollusks were not found in the diet of this species. So moving on to our discussion, or what this means, here we see the comparison of the two relating back to our original question. And we can see that in the diets of the southerns and the whiptails, some of the largest differences are that the whiptails are eating a lot more worms than the southerns. That's that 63% figure you saw earlier. And they're nearly 4 times as important in the diets of those whiptails than the southerns. For crustaceans, the southerns are utilizing crustaceans twice as often, or they're twice as important in the diets of the southerns than the whiptails. And again, teleosts were almost non-existent in that of the southerns, but were prevalent in that were prevalent in that of the southerns and nearly non-existent in that of the whiptails. Here's the life stage comparison of the southerns between juvenile and mature, and we can see that the mature are eating a lot more worms, while the juveniles are eating a lot more crustaceans. Again, mollusks were pretty much insignificant for both, and teleosts were a bit more abundant in the diet of the juveniles. So now going on to interpreting this, that's really great data, but what does it mean? Using a using a foraging strategy analysis chart, this chart here, the Costello method, we're using actually an adaptation of it. So the main difference is that we're using percent prey abundance instead of percent abundance, as it's just a bit more specific and therefore applicable to our study. So here we can see for the southerns, there are the crustaceans in the upper right-hand corner, which are a little more dominant or generalized. And then for the whiptails, we can see that the worms are a little more in the upper right-hand corner, again being more dominant and generalized. Through both of these strategy analysis charts, we can conclude that these rays are generalists, meaning that they do distribute their diet over a number of different prey taxa. However, they are partitioning in that the southerns were a little more towards crustaceans, while, as we saw, the worms were more abundant in the diet of the whiptails.
So what Luke just described about these two species being generalists is important towards our study because we've been able to recognize that they're able to adapt to their environment, whether that be short-term effects such as seasonal variance or more long-term effects such as anthropogenic effects, which is basically the impact that we as humans have on these species. And this connects to our further original objectives of how do these two species thrive and survive in their environment without one outcompeting the other, and how can they share this environment. So we've been able to try to understand this through dietary partitioning, and through the data that we've collected, we have been able to understand that they do partition their diet and they do share these resources in their environment, which is very significant towards our research project. And additionally, we also found ontogenic differences, which, as Patrick described, is the difference in life stage between juvenile and mature, which can go towards further research studies of how can we further conserve juveniles in nursery areas compared to the matures in their specific habitats as well. And throughout this research study, it's also important that we must address possible bias or error that could have been involved in our project. So first, in the digestion of prey taxa that we collected, the hard exoskeletons of organisms such as crustaceans are a lot more difficult to digest compared to soft-bodied organisms such as worms. And this could affect what we saw in our lab work analysis from the prey taxa that we collected. And secondly, although we're very confident in the method of gastric lavage, we can't be 100% sure that this empties all stomach contents of the species without actually physically going into the ray's stomach and analyzing if it's empty or not. And so future studies could be one study of anthropogenic effects of the whiptail, because as Luke described, we have analyzed ontogenetic differences in the southerns between the juveniles and males, but it would also be interesting to look at the whiptail and see if there are any ontogenetic differences in diet for those species as well. And overall, the data that we've collected has gone towards conservation efforts because the more that we know about the species and their role in the environment can go towards the conservation efforts to help the ecosystem as a whole. This is our literature cited. We would like to thank the Island School and Cape Eleuthera Institute, as well as Kate Kincaid and Dr. Owen O'Shea. Our team interns, Reed Webb, Alejandro Molera Arribas, Sarah Weiss, and Julia Cardoso. And finally, our advisors, Casey and Tanya, for not only putting up with all of us this semester, but for being amazing role models and mentors, and none of this would have been possible without you. So thank you so much. So thank you so much for listening, and at this time we'd love to open the floor for any questions you may have.
Did anybody get stung?
What's the question? Sorry?
Anybody get stung?
So the question is, has anybody been stung? So to answer your question, that's a great question. Although we're not going to lie to you and say that this isn't dangerous work that we're doing, but while it is, safety is our #1 top priority, not only for our team itself, but for the animals. And being stung by these animals, although maybe talked about or maybe in news, actually is very rare. And as Patrick explained, we take great caution by wrapping the barb that's on the tail, that's the actual part of the organism that would sting. We wrap it in a wet cloth and secure it with Velcro to make sure that that's not a danger to our team or to the stingray itself. So, thank you. That's a great question.
Yes. Why do you think that southern stingrays rely heavily on crustaceans and caribbean whiptails rely more heavily on worms? Is there an explanation for that? So the question was, do we have an explanation for the specialization in the diet with the whiptails more towards worms and the southerns towards crustaceans? So in my opinion, although we did observe some overlap, specifically in page flats, we caught both the southerns and the whiptails. They are utilizing some form of, in my opinion, spatial partitioning. And so the worms could be more abundant in the regions that these whiptails are in, and the crustaceans similarly could just be more abundant in that of the southerns. But that was a great question. Yeah.
Yeah, going along with that, have you ever encountered these two different rays in the same area? Because I'm wondering, are they petitioning for resources on any other ecological areas?
So the question was, have we seen these rays in the same environment, any overlap there? And if so, can we allude to any other partitioning of resources in these two species that are so ecologically similar?
So, while we have observed a little bit of overlap in the two species, it's been fairly rare. Like one of the times, for example, we've seen a Caribbean whiptail and a southern stingray in fairly close proximity over near Page Creek, but those times have been fairly rare. We found most of our whiptails in creek systems, such as Deep Creek, and our southern stingrays in more open ocean sand flats, such as the Marker Bar out there and Bottom Harbor, for example. So that difference in space where we find the rays, it's a very possible sign of spatial partitioning. For this study, we looked at dietary partitioning with an official methodology, and those results support that there is a possibility of dietary partitioning, but it's a very possible future study to see whether or not spatial partitioning is occurring between the two species.
Were there any surprises whether for what they were or weren't eating with the different taxa in the diet of the stingrays?
So the question was there any surprises within figuring out what rays were and weren't eating in their diets?
So these stingrays are data deficient, meaning we really don't know that much about them, including what they eat. So honestly, everything was kind of a bit of a surprise. You know, we were doing the study to figure out what exactly they were eating, and yeah, so that's what we were looking at.